Now that we have a background on free energy, chemical reactions, and ATP, let us talk about the natural catalysts that our cells use in order to drive chemical reactions. We call them enzymes. Here are some of the characteristics of enzymes. First, you only require a small amount of enzymes in order to make a certain type of chemical reaction happen. Number two, enzymes, like all catalysts, are not used up or altered when they hasten the reaction. Number three, the enzyme itself cannot affect the thermodynamics of a chemical reaction. So this third point here is very important for you to understand. So enzymes may only catalyze reactions that have a negative free energy change or that are favorable. They can never catalyze unfavorable reactions. And lastly, all enzymes are proteins. But there are certain molecules like RNAs that act like enzymes. But again, all enzymes are proteins. So an organism such as this dog or such as yourself might feel relaxed right now. You might be resting, you might be lying on your bed right now. But at the cellular level, you are actually very busy doing millions of chemical reactions. And these chemical reactions are the reasons why you are alive right now. But because of the enzymes being so efficient in running these chemical reactions, you don't feel stressed at all. In fact, if you feel your forehead right now, you feel the warmth that's emanating from your body as it does these chemical reactions. With the absence of enzymes, you would need a lot more heat in order to drive these reactions, and thereby causing your cells to produce excessive heat and causing their death. So thanks to enzymes, we are able to do very important chemical reactions without even raising our temperatures beyond 37 degrees Celsius. Here's a common enzyme reaction that we often see when we put hydrogen peroxide onto our wound. And that's the chemical reaction here is the decomposition of hydrogen peroxide into water and oxygen gas. So our wounds would contain many ruptured cells, and our cells actually contain an enzyme called catalase. Okay? This enzyme, once it gets in contact with hydrogen peroxide, can accelerate the decomposition of hydrogen peroxide into water and oxygen a million-fold, a million times faster. Thus, we see the bubbles forming really fast as the hydrogen peroxide gets in contact with our open wound. So enzymes actually have that ability. They hasten reactions. They are sort of multipliers on the rate of reactions. So here are examples of enzymes on the left and the time taken for the chemical reaction that they catalyze without them on the second column. And on the third column will be the number of reactions that each enzyme molecule can do at a certain amount of time, that's per second. And on the last column will be the rate enhancement that the enzyme would result in the chemical reaction, with the chemical reaction that it catalyzes. So here's an example, staphylococcal nuclease. Without this enzyme, the chemical reaction that it actually catalyzes, which is the breaking down of nucleic acid, would take 130,000 years to happen. That's a very long time to wait. Okay? But with the presence of the enzyme, one enzyme molecule may actually catalyze 95 of these reactions in 1 second, and that's just one enzyme. So imagine if there are 1,000 enzymes, they would be able to catalyze 9,500 of these reactions per second. And that actually results at a multiplier of 5.6 times 10 to the power of 14 times faster of a reaction than without the enzyme. So that's how powerful enzymes are. So how do enzymes work? It has something to do with lowering the activation energy or the energy required to overcome the activation barrier of a reaction. Okay, so here we have a chemical reaction of glucose and ATP. So this is a chemical reaction that happens during cellular respiration or during glycolysis portion of the cellular respiration process. So we see here the initial state somewhere in the middle of glucose and ATP, which are the reactants. Okay? And they will eventually, after this reaction, become glucose 6-phosphate and ADP, the products. And notice the change in the free energy of this reaction, which is negative 4 kilocalories per mole. Meaning to say this reaction is favorable, but without the help of an enzyme, the activation energy would be quite high and the reaction will take a long time to complete. So without the presence of an enzyme, the activation energy, as shown here, highlighted in blue, is quite high. Okay? So the transition state, the state when the substrates, we call the substrates, glucose and ATP, are held together very closely by the enzyme, is the transition state. And it will be very difficult to attain that without the enzyme. Again, notice the activation energy that's highlighted here in blue, that arrow that's quite tall. With the presence of an enzyme, this activation energy, okay, this activation barrier is lowered so that it only takes a little amount of energy now in order to reach the transition state, which was supposedly originally a lot higher than with the presence of the enzyme. So you now have this blue curve as opposed to the red curve. Okay? So because of this, it's now easier for the glucose and the ATP to reach their transition state, and therefore easier for them to react together now to produce the products. So this is how an enzyme works. Again, notice that this reaction was originally favorable, and enzymes can only catalyze reactions that are favorable. Another effect of an enzyme, as shown in this graph, is that it allows the capability of molecules reacting, okay, to be enhanced even without increasing the temperature of the molecules. Now, normally, we can only drive a chemical reaction to happen if we increase the rate of movement in the molecules so that they will collide more with each other. And how do we usually do that? We increase the temperature of the system. So notice here, without the enzyme, you would need to increase the temperature of the system up to 75 degrees Celsius in order for a chemical reaction to happen. However, with the presence of enzymes, you do not need to do that. The enzyme simply allows chemical reactions to happen even at 25 degrees Celsius. Notice here the blue highlight. Okay? So again, as I mentioned a while ago, our bodies have a maintaining temperature, wholesome temperature of 37 degrees Celsius. And even at that low temperature, thousands or even millions of chemical reactions are happening in our cells. And why is that possible? That's because our cells contain hundreds of enzymes in them in order to catalyze those reactions. Without those enzymes, our bodies have to increase its temperature perhaps maybe up to 75 degrees Celsius in order for some of those chemical reactions to happen. And we know that that is fatal to our cells. Having a fever of 40 degrees Celsius or higher is fatal to our cells because this causes the enzymes to denature. In other words, they destroy the proteins that make up the enzymes. And with the absence of our enzymes, our bodies now are incapable of making these chemical reactions happen at the wholesome temperature of 37 degrees Celsius. So as we can see in the arrow here, the enzyme decreases the minimal energy required for molecules of a chemical reaction to actually interact, thus lowering the activation energy.